Hi, everybody. Welcome to another Chats with Chaudhry. I hope you're safe and well. It was a lovely, bright, sunny morning here in Surrey, but it's starting to get overcast now. But hopefully, my guests they will brighten up the rest of my day. I'm delighted to be joined by Cathy Frankis, who's senior scientist for the Functional Ingredients Lab, and Vincent Thibon. I didn't check this before, Vincent, so I hope I pronounced it correctly. Senior scientist, pharmaceutical development, and they're both from Reading Scientific Services, or RSSL for short. And today, we are talking about vaccine, excipient, and buffer analysis, and in particular, the applicable techniques and equipment for this and the process of method development to successfully analyze buffer components. That's a long sentence. I'm about to take a deep breath now. So, Cathy, Vincent, lovely to see you both. How are you?
Hi, I'm good, Risen.
Good, lovely to see you.
Yeah, good, Risen. Thanks for having us.
It was lovely to meet you both. Hopefully it's sunny in Reading today, though, because it certainly was sunny in Surrey, but it's got a bit cloudy now, as I said. No, a bit
I'm
overcast.
afraid, no.
We're not too far away
Overcast.
from you, are we, in Reading? Let's be honest. We probably share the same weather pattern.
Well, I was hoping you might have some more localized weather, which could actually be a bit better, but hey, never mind. So before we start talking about vaccine, excipient, and buffer analysis, would one of you be kind enough to give me a quick overview of RSSL, and then could each of you give you a quick background in terms of your own backgrounds, experiences, and business, the labs you work for?
Sure. Okay. I'll go first, Rizwan. So RSSL stands for Reading Scientific Services Limited. We're based on the University of Reading campus in Reading. We're a contract analysis lab offering testing and pharmaceutical testing and for pharmaceuticals and food products, and also research and development for a number of clients. We've got a whole range of capabilities and technical expertise. Yeah, we've been here for about just over 30 years now, I think. Kind of a quick overview of RSSL.
It's always difficult when you have to talk about your own company, I know. So, and what's your background, Cathy, and what's the lab that you work for?
Sure. So my background is I'm a chemist by training. I did my PhD at Bath University a few more years ago than I would like to think. So I'm an inorganic and polymer chemist for my PhD. I joined the company about 10 years ago. I actually started off in the sales team here, which gave me quite a good insight as to all the different techniques that we offer our clients and how we problem solve. And that then led me back into the lab to the investigative analysis team. So I spent about 6 years. And then a couple of years ago, I then moved up to our functional ingredients lab, where we specialize more in chromatography-based techniques to method develop and perform routine testing for both food and pharmaceutical clients. So what I'm doing now is I'm actually just started a split role between the two teams. So I'm senior scientist specializing in both chromatography and spectroscopy techniques. Yeah, this has been a little bit about me.
Fantastic. And Vincent?
Hi, so I performed my PhD studies in Leeds, again, quite a long time ago, analytical chemistry in capillary electrophoresis. And throughout my career, I have been first working in consumer healthcare for one of the big pharmaceutical companies, moved and worked with chemists, early stage project, and then worked for the leachable and extractable group before joining RSSL last year. So I've only been here a year. I'm an analytical scientist, mainly HPLC, but also doing gas chromatography. And the department that I work in at the moment, pharmaceutical development, offers many different activities encompassing method development, method validation, method transfer. We do assay testing, we do stability indicating and related substances methods. We look at drug product, drug substance, we offer dissolution, we have stability chambers, so we perform stability testing. We can also store stability samples for clients and ship them back to them. we interact with other departments when we don't have the technology. So we, for example, have mainly HPLC with several detectors, UV, RI, fluorescence, prep HPLC. But when we don't have, we go to places where we can find the ICP-MS, triple quad, NMR, which gives us a good array of technique to use to satisfy the inquiries we get from clients.
Fantastic.
So
Fantastic.
here it is. I don't know. I can go on.
No, no,
I'll
that's fine.
stop
But that's a great overview. So thank you. So let's start with the first question. So why is it important for manufacturers to have confidence in the component concentrations of their buffers?
So I guess from a kind of an overarching point of view, you know, any manufacturer needs to be able to show that what they think is in a product is in a product. Maybe they are outsourcing the manufacture of a particular thing and they need some confidence that the outsource manufacturer has followed the recipe. Maybe they're manufacturing themselves and they need to show that the recipe has been followed internally and that it meets the specifications. So it's always important for manufacturers to have the confidence that the components are in the products that they think it is, and also that they're there at the right concentrations. And with respect to buffers, these are normally quite simple solutions really compared to some of the matrices we work with here, especially on the food side of the business. Buffers are incredibly simple. They're essentially just aqueous solutions of salts and mainly, well, organic and inorganic components.
Right.
The reason for why they're there is to maintain a stable pH normally, and to prevent microbial growth, to promote the stability of the active ingredient that buffer is going to be used to solubilize, and to just make the product as biocompatible and suitable for use as it's intended, really.
Right. Okay. And Vincent, do you have anything to add to that?
No, I was going to say something until Cathy just
She's so thorough.
said it all. No, I think she's doing a very good job.
Fantastic. And what general methods can be employed for the analysis of vaccines, excipients, and buffers?
So I think normally we would be, I mean, they're quite, as I said, they're very simple matrices. So we actually don't necessarily need complex separation techniques, but maybe more for but there's always going to be instances where ingredients could potentially interfere with each other. So
Right.
separation techniques that involve some kind of separation of the different ingredients are important. And so mainly that's chromatography based techniques. So HPLC with array of detectors, UV for potentially components that have got chromophores. We're going to talk about ion chromatography, so that would be used for components that have a permanent charge. Or HPLC ELSD is another method of detection that can be used following liquid chromatography. If we're thinking about inorganics, potentially there might be a requirement to use ICP OES or AS, which are techniques that our metals lab offer.
Right.
And quite often it will be a combination of techniques, maybe some chromatography, but there may be also some metals-based techniques to look for and quantify the different components that should be present.
Right. Vincent, anything to add to that?
Well, I would say the method that are going to be employed are there's the separation techniques that Cathy's talked about. Some of the things I can think of is how to prepare the samples. Some of the samples are going to be able to be detected with one detector. Some others will not have any chromophore groups to be analyzed with the same detector. So there are prep techniques. We can derivatize, we can make them UV responsive, and all of these are techniques that can take time. They're probably going to limit the applicability of one particular separation. So I think it's depending on the actual components. So in the case that we were going to talk later, the components we were dealing with, some of them are going to be present in low amounts, very low amounts compared to others, and will require some kind of treatment. There's different techniques that have been reported, but for some of the components that we dealt with, there were very few analysis. I can think of thin layer chromatography, which is an old technology, but yeah, we can in the case that we dealt with, we had three components, and as Cathy mentioned it, we have to separate them in the first place to be able to then quantify and give the answer that Cathy was talking about, identity and quantitation.
Great. Okay, thank you for that. So you mentioned ion chromatography a bit earlier, Cathy. So how does ion chromatography help in the analysis of vaccine excipients and buffers?
So ion chromatography is a field of chromatography that I've come to love over the last couple of years. I had the opportunity to, I might mentioned earlier, to move to our functional ingredients lab and run the ion chromatography service offering while my colleague Maud was on maternity leave. So it was a real in a deep end experience for me, but I very much enjoyed it. And ion chromatography is essentially a chromatographic based technique where permanently charged components are separated based on their charge, their affinity, their charge affinity essentially. And rather than using different concentrations of organic to elute components off a column as you would do in reverse phase HPLC, we use the hydroxide concentration. We increase that over the course of a run in order to elute variously different charged anions from a ion exchange column. And the main sort of application in terms of buffers would be using conductivity as a detector. So yeah, I mean, the reason why it's important for buffer analysis is because a lot of the sort of salts and inorganic components that are present, such as chlorides, phosphates, sulfates, they are all very easily analyzed in the same screen, in the same injection we use when we use ion chromatography. It's yeah, which is obviously really, really handy. We can separate using that gradient, and using the right columns, we can get them into quite a short runtime.
Right. Are there any major sort of challenges to you though when you use ion chromatography in that process?
Well, I think as per all chromatography-based techniques, you can run into potential issues if you have interfering components. So an example could be I mean, EDTA, for example, we have experienced some issues where that can co-elute with the later eluting anions, such as phosphates. And if you can't separate those off, that can then cause interferences, which can affect the accuracy of your data. So you have to make sure that you have the specificity, really. The method that you decide to choose for separation, you have to make sure that it then doesn't interfere with anything else that could be present in that particular buffer.
Right. Okay. Thank you for that. So, Vincent, let's talk about the pharmaceutical development lab. So, how else are you able to help people within the COVID-19 supply chain?
Well, we can, as we've demonstrated with this particular project that we did, we are rapid to react, especially that the deadline when we from I would say kickoff meeting with a client to the supplying of the data, we had less than a month, and that included doing the literature search, ordering the column that we needed for the analysis, to performing some kind of verification on the method so that we could have confidence in the data that was generated. So I would say is that we are versatile. We can quickly switch from one thing to another and apply our expertise. So I have been involved with this very particular project, so I think I'm going to talk about this very particular project. But we were faced with a client that was employing somebody else to perform the analysis, and they were not able to separate one of the compound, and they came to us to, not begging, but saying we need to have these results in a month. And so it was quite challenging. As Cathy said, we were faced with components that had, one of them had a UV chromophore, but the other two, one could not be analyzed by UV. One had to be derivatized if we wanted to. But doing a literature search, we came up with a column that could separate all three components. And to avoid having issues with the detection, we decided to go for a detector that was mentioned previously, which is ELS detector. And so in the same run, in a very quick analysis, it involves different dilutions because, as I mentioned, the components are present in different order of magnitude. But we've applied what we apply to every project is literature search, quite a rapid method development, a bit of luck, and also all our effort to deliver on time. So I think if we had other projects, we would perform the analysis the same way.
Fine. Great. So there's one question I forgot to ask, is what problems, issues can be faced when attempting to analyze buffer components, and what is the general approach to method development to overcome some of these issues?
Cathy, shall I go on?
Yeah, that was for you, Vincent, actually.
Okay. Well, I think I've alluded to some of the problems that we faced. I think another issue is with the particular techniques that we used, we went for a column called Helix column, which is best suited for separation involving, well, it needs a lot of organic solvent to behave in the way the Helix mechanism happens. And we are trying to inject on the column a sample that is effectively 100% aqueous when we get it. And because of several reasons mentioned, the low concentration, it wasn't possible to do much with the sample itself. So we had an issue of mismatch between mobile phase and sample. We had an issue where two of the components had a very similar behavior due to one of the groups on the molecule, which was the reason why the initial lab that was commissioned to do the analysis couldn't perform it. It's only when we had the detector allowing us to see all components in the same run that we were able to separate them. As Cathy mentioned, it's almost like having specificity. I was tasked with the analysis of one of the components specifically, and I had another of these components eluting next to it or right beneath it, and that's what was actually detrimental to the analysis in the first contract lab organization. And it's the availability of having that sort of instrumentation that allowed us to have a successful outcome. So yes, pH issue, we had to shift components during the analysis, dilutions, detection. What else could I say that I faced? No, that's pretty much it.
Great,
on top of the sort of tight deadlines.
Thank you. Okay, so that's all we've got time for. So where can people get more information?
So there's quite a lot of information on our company website, so www.rssl.com. I believe there is a specific section of that website dedicated to buffer analysis with respect to how we might be able to support clients who are trying to develop things in response to the COVID situation.
Sure.
You know, Vincent referenced there to projects where we've had to work really, really quickly. And so we are really keen to harness the expertise we have to help our clients move quickly wherever we can. So there's a lot of information on the website, which will link to some case studies. But also we're quite vocal on the old social media. So if anyone wants to follow RSSL on LinkedIn, then there are lots of updates there.
Fantastic. Well, all that's going to say is thank you very much for your time today, Cathy, Vincent. It was lovely to meet you both. As Cathy just mentioned, there's lots of information on the RSSL website. You don't have to write anything down. The links will be above the video, so you can click on them straight away. And all that's going to say is thank you both for your time today. It was lovely to meet you both. It was great to hear more about what you're doing. I wish you a fantastic, happy, and more importantly, healthy 2021 and a fantastic summer. So thank you very much for joining me today.
Thanks
Thank you very much.
Cheers.
No, thanks a lot.
And thank you for watching as always. And until next time, stay well and stay safe. Bye-bye.